Hello, everyone. My name is Noah Higgins at Truman State University of Kirksville, Missouri, and my topic today is a brief review on chronic wasting disease, as well as a focus on its prevalence in non-cervid organisms. So let's go ahead and get started. So first and foremost, it's important to look into why this is important. And just as a little bit of background, chronic wasting disease is a prion disease, and these diseases possess a 100% fatality rate with no known cure. And chronic wasting disease specifically is a major concern because it's a disease that's expanding very rapidly in both wild and domestic deer populations. And this is primarily due to the fact that the infectious agents, the prions, are able to persist in the environment for extended periods of time. Now, as a result of this very difficult controlling effort for the disease, it has multiple ecological and conservational concerns, mostly dealing with preventing deer from being infected. However, up to this point, until very recently, the disease was believed to be exclusive to cervid organisms. However, recent evidence has suggested that it is potentially susceptible in other organisms as well. So, just a little bit of background about what prions are. So, prions refer to misfolded proteins that are spreading their misfolded structure. In this figure here, we see the traditional expected folding of a prion protein. This is the PRPC folding. As we can see here, it consists mostly of alpha helices, and this is what we want to expect in these proteins. And currently, it's not understood what these proteins are responsible for in their PRPC function, or their folding, I mean. However, there are multiple theories that currently exist with people's guesses ranging from their involvement in cell growth or cell proliferation. Now, prions are interesting because they are currently the only infectious agents that do not possess any DNA or RNA. Rather, they're able to spread through their self-propagating structure. And what this means is that once they're misfolded, they're going to essentially reach out to these other PRPC proteins and cause them to reconfigure themselves in a way that matches the PRPSC folding. And this altered structure, as we'll get into later, is what leads to a increased risk of disease. So these prions are able to cause disease due to this chain reaction folding process. As I said earlier, they sort of instigate the transformation process, and this is done by the formation of a prion nucleus, which is essentially just a grouping of multiple of these misfolded prion molecules put together. And once this nucleus is assembled, this enables it to sort of travel around and convince the PRPC that it is incorrectly folded and needs to reconfigure itself to the PRPSC configuration. So as we can see here, this is sort of what we were seeing in the previous slide with the alpha helices model. However, the PRPSC, which is the prion infective version, it has this beta pleated sheet here instead of a third alpha helix. And this is what really causes most of the problems. Due to this beta pleated sheet, aggregation is more likely to occur, so there's going to be a lot quicker buildup of this protein, essentially blocking neural pathways, leading to neural degradation and deformation, and it's just going to gather up and cause problems neurologically. It's going to disrupt your brain function, and eventually this leads to permanent damage, included but not limited to ceasing of neuronal function, and ultimately leading to death. So what is chronic wasting disease specifically? So chronic wasting disease is one of these prion diseases, and as I stated earlier, it primarily just affects cervids, which is the family of deer, moose, and elk. And as with all of these other prion diseases, these experience a 100% fatality rate. And this is a cause for concern because this is currently shown to be the only prion disease which is currently found out in the wild. And it has expanded very widely geographically since it was first discovered in 1967. So here we have a little bit of a timeline of the spread of chronic wasting disease. So as we can see, it was first discovered in 1967, and just as time goes on, it was found in more and more species until we get to 2000, where it was first reported in South Korea, because prior to this point, it was only experienced in America. And so this is why it's kind of a growing concern, because it's reaching further and further away from the point of initial infection. So let's take a look into the spread of chronic wasting disease. As we can see in this figure here, it's spread very, very widely throughout the US. And the disease is primarily spread through multiple means, but primarily is the direct contact between an infected and uninfected animals, as well as the indirect spread through infected bodily fluids. And the bodily fluids is what really causes a lot of difficulties with conservation efforts, because even if an infected organism is killed and disposed of in a way that won't result in more infections for other animals, it still is going to have left multiple reservoirs of infection, which are caused by it defecating or urinating or even bleeding in the environment. And once these bodily fluids are spread, they're going to contain the prions, which means any organisms that come into contact with this infected bodily fluid are going to potentially take in the CWD prion and as a result start carrying it as well. Now, soil specifically has been shown to be a very good sort of host, I guess, for lack of better terms, for the prions. It's able to retain them very well for extended periods of time, and there are multiple transmission pathways. So no matter where you intake it, it's going to be able to find its way into the central nervous system and eventually reach the brain. So here we can see its impact on deer populations. As we can see by this figure here, CWD has a major effect on the death of deer, and it is a major source of death in both domestic and wild deer populations. As a result, it causes severe behavioral decline as they get more and more infected, and ultimately it alters the ecosystem dynamics because deer that are interacting with one another more after being infected are going to result in more infections and thus more death. And additionally, because susceptibility of chronic wasting disease is rooted entirely in the PRNP gene, so if they have that gene, they will be susceptible. This places a sort of natural selection on the genetics of deer susceptible to infection. And this ultimately brings us to the focus that I want to talk about, which is the potential for cross-species transmission. Now, while this is a relatively new sort of field of study that's opening with chronic wasting disease, there have been multiple recent cases that have shown the potential of cross-species transmission of the disease. In some of these cases, the prions have been shown to be able to adapt to their hosts as well, and as a result of this, there's a sort of uncertainty that exists around the species barrier. Prior to this, we thought that it was pretty concrete and that the disease was only going to affect cervids, as it had only been affecting cervids due to the PRNP gene. However, due to these more recent studies, it's sort of raising uncertainty. And ultimately, as I said earlier, this sort of depends on the compatibility of the protein and the strain type. So the strain type might react differently to PRNP than other strains, but this altogether is going to raise concern of the potential spread of chronic wasting disease into new and different species. So first off, we have a study in which deer mice were inoculated with chronic wasting disease prions, as well as Creutzfeldt-Jacobs disease prions, which is another prion disease observed in humans. But we're going to focus on the chronic wasting disease one specifically. In this instance, the deer mice showed successful infection of chronic wasting disease prions as shown in this figure here. It's just in different regions of the brain. And following the inoculation, 100% attack rates were experienced, and this serves as a strong sign of evidence about potential adaptability of prions, having a high transmission rate of 100%. All the mice were inoculated, and all of them received the misfolded structure. And they also experienced very similar incubation periods and patterns to what's been observed in cervids. So next we have an example in which ferrets and raccoons were inoculated with chronic wasting disease prions. This top figure here is going to be from the study in which ferrets were inoculated, with this bottom figure being from the raccoons. And as we can see here, the misfolded prions are conglomerating and are observed in both of their brains. However, interestingly, prions developed different strain differences when they were compared to the strains that were initially infected. The raccoons served as a sort of intermediate species, and following their infection, they developed a different strain configuration than what had been observed in deer. However, despite this, the strain that was now in the raccoons could still be transmitted back to the deer. And this is interesting because this presents the possibility that intermediate species can serve as a sort of breeding ground for these new strains of proteins as they adapt and change in intermediate species. Now, all this is to say, this is still a major environmental risk, and the reason that raccoons were used in a prior study was because they have a key role in a lot of ecosystems as scavengers. So they're usually coming into contact with corpses and carcasses of potentially infected deer. So if they're able to uptake this infected prion, then it's much more likely that they could carry it to another deer and spread the disease to another population as a result. And this is a major concern because, as I've stated multiple times, these prions are able to persist in the soil for extended periods of time. And this is only made more difficult by how hard a lot of these habitats are to manage. And this is because there's a lot of variables that go into where sites of infection may be. So in one study we've seen here, it was tested how many deer were infected with chronic wasting disease around a central set of feeding blocks. Now, keep in mind, these sites are very frequently sites where bodily fluids such as saliva are going to be exchanged due to these feeding blocks. And as we can see, these variables also impact the distribution of which the disease is found. But even then, it's not uniform. And ultimately, this only gives rise to another problem with the fact that it's also extremely difficult to remove and neutralize infected prions that are already in the soil. These infected prions have been shown to be resistant to a lot of industrial grade cleaners and disinfectants, and so it's very expensive to go about cleaning them. So I've spent a good amount of time talking about the different strains, and currently we know of 10 chronic wasting disease strains that are proven to exist. And these strains vary just in structure, as well as with slight variations in behavior and incubation time. They don't really show that many major differences from one another. And as we can see here, these are the 4 most common strains in North America. CWD 1 and 2, 116AG, and Wisconsin 1. All of these other areas are either hodgepodges of strains or areas where a clear dominant strain hasn't been identified yet. And ultimately, these strains have been shown to evolve in their hosts, as we saw with the raccoons, and potentially new traits can emerge from this evolution, such as the development of prolonged incubation times. And as we saw earlier, there is the potential that non-cervid hosts may further alter the disease. Now, this brings us to the ultimate question of whether or not there is a potential for zoonotic disease as a result of chronic wasting disease. And while there are currently no confirmed cases of chronic wasting disease being able to alter the prion protein in a human brain, it has been shown that a lab conversion is potentially possible with the elk strain of chronic wasting disease. And ultimately, there is a precedent for these zoonotic diseases for prion diseases, as mad cow disease, which is another form of a prion disease, has also been shown to have effect with humans. And ultimately, the risk for this is only going to increase over time as the disease spreads and develops more, so it's going to be important to continue monitoring this. So now the question is, how can we manage this? What can be done about this? And ultimately, there's a lot of challenges that exist. So culling infected deer has primarily been one of the main ways that departments of conservation have tried to fight the spread of the disease. And a study in Wyoming, which this figure is not from, but a study in Wyoming showed that while these culling efforts do slow the spread of disease, they don't outright hinder it that much. So it's sort of an uphill battle. And as I've said a million times, the environmental persistence of these prions is really the main challenge. Even if you were to take out all the infected deer, the infective agents can remain in the environment for very long periods of time. And additionally, these prions start shedding before symptoms can emerge. So even if you think you take out all the infected deer and prevent them from spreading infective agents, there's still the chance that there are infected deer shedding these prions beforehand. And another thing, I haven't mentioned it yet, but the disease is also very easily vertically transmitted. As we see here, the misfolded prions spread all over the body, but as here we can see, the uterus has plenty of them. And so ultimately what this means is that infected mothers will often pass down the disease to their offspring. So ultimately there's a few future concerns for this. There's an increasing number of hosts with different species from non-cervid families, which can potentially lead to more chances that the disease can further mutate and develop. The increasing number of hosts just outside of just even with the only cervids, is creating more reservoirs of infection, resulting in the disease staying more prevalent. And the more the infection spreads, it's going to be more and more difficult to contain. So these problems are only going to multiply upon themselves. And as more strains develop, there's going to increase a risk of trans-species infection. So in conclusion, it's important to realize that chronic wasting disease is expanding at an alarming rate, and evidence supports the idea that it might not be only a cervid exclusive disease in the foreseeable future. Strain evolution is real and is occurring, and it should be a concern for people. And so as a result, more research of the disease's range and continued monitoring of non-cervid hosts is important. Attached are my sources. Thank you for your time and have a great rest of your day.